Good afternoon. I'm very pleased that today we have Dr. Bernard Rabin from UMBC to give the Jim Joseph Memorial Lecture today. Bernie and Jim go way back. I even had to ask him last night, well, what year did you actually meet? Because ever since I've knew Jim, I knew Bernie. And he said that they met in 1980 at AFRI, which is the Armed Forces Radiobiology Research Institute. I said, wow, that long ago, really? And since they met and worked with radiation, eventually that led to their grants with NASA, in which they looked at the effects of particle irradiation on age-accelerated markers, particularly in the brain. And at that time, no one had actually looked in the brain to see what was going on in the brain. They thought, oh, radiation won't affect the brain. But as you'll see today from his talk, that wasn't actually the case. And I met Bernie when I started, not long after I started here in 1996, and we've been collaborating on our NASA grants ever since. And he actually, I looked it up, I counted, he has 41 papers that he's published with Jim over the years. So I think you'll get a good feeling for the research that they did together from the talk today. So at this point, Samin's going to come up and present Bernie.
So it's really a great pleasure to have this special symposium and presentation in honor of Jean, and I would like to recognize Marlene, Jim's wife, who is a good friend, and it's wonderful to have her come for these lectures as well. So I don't think I need to tell you very much about Jim, but he certainly was a wonderful and an important investigator at the center, and someone who you would recognize and distinguish right away just because of his own personality and the way that he really was very down to earth and would say it like it is. And over the years, we had developed some good relationship, and he certainly, the work that they've done with Barbara and with others and other of his collaborators have certainly made a big difference in terms of bringing attention to the role of nutrition in the aging brain, and I know that it has certainly made a difference in sale of blueberries. As a result, I can tell you that now I myself have 3 blueberry bushes in my backyard, so you can say that he's made a big difference. So it's now my great pleasure to invite Dr. Rabin, our today's speaker, so that we can present him with this plaque that was made in the honor of today's lectureship and before his presentation.
Thank you very much. I appreciate it.
Thank you so much for coming.
I really want to thank Barbara and everybody else for inviting me here. As a consequence of that, I'm doing something that Barbara has never seen since we started working in 1996. And that is I put on a tie. So I really do want to thank you for the honor and very much for the plaque. I do appreciate that. So what I'm going to do here is deal with some aspect of the research that Jim and I did and then that Barbara has been working with me on, or I've been working with her on. And that is, first of all, to give you an introduction to space radiation, what it is and how it works. Then we're going to look at some of the cognitive effects of exposure to this type of radiation, how this relates to aging, how it relates to oxidative stress, and finally to dietary countermeasures on this. So this is where we are currently. Low Earth orbit. This is the International Space Station, the space shuttle. But what we're talking about for future trips is to Mars and probably even beyond Mars. And the environment there is very different than on Earth and in low Earth orbit as well. This gives you an idea of what this space radiation environment consists of. There are particles of high energy and charge, iron, titanium, silicon, you name it, it's out there floating around. You name it, it's going to produce effects. And that's what we've been studying. And this one here gives you an idea. The Earth really does a great job protecting us because these particles are charged. They're kept away from the Earth, except that there are trapped particles around, the so-called Van Allen belts. But that's not where we work. We leave here, go to Mars or to any other place. We're going to be exposed to these types of heavy particles. And we've got to know what they're going to do to astronauts. What I'll be telling you is not going to be a mission stopper, but they should know what they're getting into. Okay, here's what we get from earthbound radiation, X-rays, gamma rays. As they go deeper in the brain or in the body, the dose decreases. The heavy particles that you see out in space are very different. The dose is relatively constant until the particle can't go any further, stopping, and then the dose shoots up and then drops off immediately, which is why they're now starting to use that for radiation therapy, particularly as it relates to the brain. Because we can make, when we know where the tumor is, we can make sure that the maximum dose gets applied there. So it's very nice there, but it also presents some problems for astronauts. What we have here is to show you the doses, the average dose that is experienced on the Space Shuttle and the International Space Station. And you see it's really pretty low. This is what they would experience at Mars. You can see it's going to be a very different environment that they're going to be working on. And when Jim and I got started, NASA was, as Barbara said, who the hell cares? The brain is radio resistant. Well, yeah, it is when you look at gamma rays, when you look at X-rays. But what Jim and I showed, along with Walter Hunt at the beginning, was that when we look at these particles, there is a very real problem associated with it. So we went to particle accelerators. And any time I tell somebody that my research as a psychologist requires the use of a particle accelerator, they look at me like I'm crazy. But now we've been working mostly at Brookhaven. I'll show you a picture of the AGS where we started. Now it's at the NASA Space Radiation Laboratory. And this is what these tunnels look like. They send the particles around in circles until they get to a certain speed, a certain energy, and then they take them out. In the old days, you can see Jim and I, you can see Jim, he's reading the paper because they ran us on a 24-hour-a-day schedule. And they were not used to having the machine operate like that. You know, they'd come in and they'd run the machine for days and weeks and years and then shoot the particles out. We're coming in every 2 minutes to change samples. Things kept breaking down. So you had to bring something to keep you company while you're in there. And it was in a big barn of a facility, and it really was not a very pleasant working environment compared to today. And you can see two techs enjoying themselves in a nice whitewashed environment. We're looking in to make sure they're not doing anything they shouldn't be doing, but what the hell. So basically then the question becomes, what are the effects of exposure to these particles on brain function and on behavior? And second of all, how do they interact with the aging process? And that question came up because of Jim's interest in aging. And we got invited to Berkeley to see what went on with, to apply our endpoints to their machine. And what we found, you can see, was this. is that exposure to a half a dose produced a significant decrease in dopamine release starting 12 hours post-irradiation, and it never got any better. 6 months later, by the way, the reason that we didn't do the behavioral testing out here 6 months later for a very simple reason. Spray dollies keep gaining weight and gaining weight, and they were just so damn fat they couldn't hang on to the wire, so we stopped doing that. So you can see here what they're doing. Jim had a slide made up at Aversion, which he destroyed. I'll never forgive him for that. Picture this, the graphic artist there, by the way, had a lot of fun with it. There's a fire underneath here, and a bead of sweat coming off of the rat's brow. When he first presented that, somebody in the audience screamed bloody murder. Jim panicked and got rid of the slide. Bad mistake. But the initial work we did with Jim was looking at motor behavior. I was looking at amphetamine and lithium chloride induced taste aversion learnings. Amphetamine is in the yellow, lithium chloride is in the blue. These are two different energies of iron. And what you see is that amphetamine is still not producing a taste aversion because the dopaminergic system is no longer there, is no longer functional. Amphetamine requires an intact dopaminergic system. So the other type of work we did, and this is Barbara and Jim, they're probably admiring the behavior of one of the rats I'll show you shortly, who was really very smart. But to some extent what we did was take behaviors that we were working on and apply them in this environment. And you can see here, control rat, after he's learned and they've moved the thing on him, he starts out, goes to where it was, and then says, oh shit, and goes to where it now is. One of these radiated animals, you can see spatial memory is shot to hell and gone. Just no effect, he's just wandering around until the time is up and they lift him out. And if you want to look at it from the point of view of a graphic presentation, this is what you've got. You can see on the second trial, the control animals are doing a hell of a good job. The radiated animals aren't. Probably up here it's because they're going around in a circle and they hit the thing accidentally. But see, there's no improvement, but they do learn eventually. Looking at it, using a slightly different task, this is helium particles. And by the way, let me point out to you something here. From the point of view of NASA, the dose to which these astronauts will be exposed is between about 30 and 40 centigrade. So the doses I'm presenting here are well within that range. These are the doses to which astronauts will be exposed. And you can see here, by the way, none of these are lethal doses. The rats will live to be right to a ripe old age. Less than 1/10 of a centigrade, we're getting significant decreases following exposure to helium particles. The other task we do is operant responding on a fixed ascending fixed ratio schedule in which the rat has to make increasing numbers of responses in order to get a food pellet. So from 20 responses per food pellet up to 35, you can see that the radiated animals do not show a normal increase in responding. You can see it here with helium particles, similar type of thing, but again you see that the effective doses are around 0.5 to 1/10 of a centigrade. NASA's got problems. Okay. Novel object recognition, this is iron. And the reason I'm showing you the different particles is so that you become aware that this is the environment out there. It's not going to be just one particle that's going to produce an effect, but it's the entire environment out there that produces this kind of an effect. Okay. Titanium, again, the same type of thing. And the thing I like to point out to people who don't know better, let's hear it for rats and behavior. They're just like our kids. If they can screw us up, they'll do so. Okay, so this is from science, from American scientists way back when. But this is what we think of when we think of space travel, right? You go out there, and as you approach the speed of light, time slows down so that the people on Earth are going to be getting older, but you're not going to be getting old as rapidly. But exposure to these particles produces changes in neuronal function, neurocognitive performance that are characteristic of the aged animal. There are deficits in dopaminergic function, cognitive functioning is impaired, spatial learning and memory, object recognition, operant responding, all of these are affected by exposure to these particles. And Jim coined the phrase accelerated aging. And that's what you get. So this gives rise to two questions that we have. Are there particle-induced degenerative changes as a function of irradiation? In other words, is there an interaction between the normal aging process and what we do when we expose these animals to these particles? And second of all, are older animals more susceptible to the deleterious effects of exposure? And that gets important because if you look at NASA's requirements for astronauts, how much radiation they can take, astronauts are classified as radiation workers. As you get older, NASA will allow you to have more radiation. Hey, but what NASA didn't take into account in developing these guidelines is that as you get older, a smaller amount of radiation is likely to produce greater amounts, greater effects. So we see that here. Okay, when we look at the animal 3 months post-irradiation, you see you need a dose of almost 2 gray to produce an effect. When we retest them at 11 months after exposure, we're getting an effect at 1 gray. Okay, with carbon, okay, the rats didn't cooperate, so I only had 3 of the 10 animals included here. But you can see if this is where they should be, 10 centigrade is a dose. 10 months later, we're getting an effect at 5 centigrade. And if we look at it across all of the stuff we've looked at, certainly starting with silicon and carbon, you can see that there's a significant decrease in the dose needed to disrupt performance. Oxygen has a bit more of a problem because the doses are so low to start with, it's hard to get any lower. Okay. Now, finding this says, hey, are we going to have an effect as a function of age? That is, if we take an older animal, is a lower dose of radiation going to produce a disruption of performance? So we looked with Fisher 344 rats, which are the standard behavioral model for this, and you can see, at least this is the elevated plus maves, yes, okay? 2-month-old Fishers needs a dose of 200 centigrade. 7-month-old, we're getting significant effects at 50 centigrade. Okay, and that carries through for 12 months, so there is kind of a bottoming out effect. So this is where the major difference occurs. One of the things we looked at was the trials to acquisition. As you get older, it takes longer to learn new material. So you can see here that for the 7-month-old animals took longer than the 2-month-old. These are Fisher 344s. We'll take a look at that later, because it looks like a little bit of radiation may be good for you, but I wouldn't want to swear to that, particularly as you start reaching my age. Okay. So this is what you've got now with it. These are rats that were Fishers that were radiated at 2 months, tested a month later, 200 centigrade. Radiated at the age of 7 months, tested at 11 months, 50 centigrade for a significant effect. At radiating at 12 months, you get a slight effect at 25 centigrade. And at 16 months, which would have been the age of John Glenn when he was sent back up into space, maybe it was good for him. I don't know. So we can look at it a slightly different way, hold the age of testing constant, relatively constant, vary the age of radiation, and you see it's clear that age of testing is a critical factor. The age of radiation does come into it as well. And if you look at it from this perspective, this is the threshold dose needed to produce an effect. Radiated 2 months, tested at 12 months, you still need up there. 7 months, 12 months testing, threshold is down to 25 centigrade. radiated at 12 months, tested 14 months, even further decrease, and at 16 months, the effects of aging are primary. The radiation can't do anything more than's already been done. So this is what we have. Age modulates the risk of radiation-induced deficits. Older subjects show deficits following exposure to lower doses of the HZ particles. An interaction is here between age and HZ particle-induced deficits, so that young rats that don't show a deficit immediately do when they're tested later. And then we get to the very old animals. The effects of irradiation outweigh, the effects of age outweigh those of irradiation. So what are we going to tell our astronauts? Well, exposure to doses that they may experience out there can accelerate aging. So the next question then becomes, how do we reduce these effects? Now you can't do shielding. There's no way of shielding. Yet what you need to shield against these particles is hydrated concrete. Try launching enough hydrated concrete to provide shielding. It ain't going to happen. But it is a real problem, and we have to figure out how to reduce these effects. And how we reduce it depends on what we believe the causes are. Now, one thing that was argued for many years was that there are microlesions. This is from Apollo 8. They took up photographic emulsions, and they saw what different particles would do to these photographic emulsions. So here is the primary track of the particle going through. Out from either side of it are delta rays. These are electrons that are scattered around the main particle beam. And so you thought, hey, well, it's coming through. Maybe what's happening is that they're knocking out cells. Okay? And they estimate that on a 3-year Mars mission, pretty much every cell on the brain is going to be hit by one of these particles, if not more. But, you know, that doesn't seem to account for it. Not present the data, but some of the data we have says you get these effects from irradiation of the body only. So it can't be those particles going through the brain. So the other thing that's been argued is oxidative stress comes into it. When we look at this, and this is work, again, coming out of Jim's lab and Barbara's lab, you can see that there are changes in free radicals as a function of radiation in the striatum and in the frontal cortex. So let's look at oxidative stress. And from the point of view of aging research, oxidative stress is considered to be one of the main causes of aging and the deficits that occur as a function of aging. So, okay. So we have endogenous sources, which is mitochondria, oxidation of dopamine, exogenous sources of oxidative stress, ionizing radiation. This leads to impaired physical functioning. You have aging, we have carcinogenesis, neurodegenerative diseases. And as we'll see, some of the work that's coming out of here by Shabu-Kulus is really providing a lot of information about the relationship to neurodegenerative diseases. And how do we deal with that? Well, given Jim and Barbara and where they work, we decided to look at, or they decided to look at, flavonoid antioxidants. So that's what we started doing. And look at this. There is a significant saving of dopamine on animals that are fed on the strawberry and blueberry diets. So that's really looking pretty good. And work by Shabu has shown that if we look at the compounds that eliminate or affect oxidative stress, superoxide dismutase, you can see that the radiation reduces it. Strawberries effectively increase it. Blueberries increase as well. Another one looking at glutathione peroxidase. Again, you can see radiation knocks it down. Antioxidant diets of strawberries and blueberries are very effective in preventing that. So how does this impinge on behavior? Well, has no effect on lithium chloride, which doesn't depend upon dopamine. See, in control animals, amphetamine does not produce a taste aversion because the radiation affects the dopaminergic system. In the animals that are given blueberry diets and strawberry diets, there is a taste aversion. The dopaminergic system remains intact. Okay. We see a similar type of thing with regard to operant responding. They're on a 2% diet for 8 weeks. You can see that control radiated animals are having an effect. The blueberry and radiated animals are normal. That one is actually rather backwards, but okay. The two, strawberry and blueberry, are actually the ones that they don't have an effect. Control doesn't produce it. The non-irradiated animals are up here, and strawberry is effective, blueberry isn't as effective. And we look at it slightly different. Here's your control, not radiated animals. You can see they're way the hell down. They don't respond well. Strawberry and blueberry animals are still responding along with the non-irradiated controls. Novel object recognition, no effects. You notice here that radiated animals are not significantly different on the amount of time they spend exploring the novel object. But when you look at the diets, here's the control non-irradiated, more time with the novel object. Both of the blueberries, this is a strawberry, chooses a significant saving. They spend more time with the novel object than with the familiar object. It doesn't matter whether it's a 2% or a 4% diet, you get the same effects. This is work by Shabu showing that you get an effect on the mammalian target of rapamycin, on the PHF-tau, and these become important for two reasons from my perspective. One of which is you see that there are differences as a function of time following the radiation, which suggests that there are multiple mechanisms involved. And this one is very important also because it seems to be involved in development of Alzheimer's disease and other types of neurodegenerative diseases, so that the radiation per se may increase the probability of developing Alzheimer's, but the diet, in this case strawberry, works very well. Again, you can see this in a slightly different format, again courtesy of Shabu. Blueberries work pretty much the same way, decreasing the phosphorylation of tau proteins, again decreasing the probability that the rats or that astronauts ultimately will be more likely to develop Alzheimer's or other neurodegenerative diseases. So, exposure to the types of radiation encountered in interplanetary travel will have consequences for the astronauts. These consequences are related to accelerated aging, and it will include cognitive deficits, which may produce problems for them getting back safely from their mission. There's going to be post-mission potential problems, increased possibility of Alzheimer's disease or other neurodegenerative disorders, again, which characterize aging. And as we see, and as you've all shown here, as Barbara's shown in that lab, that the dietary countermeasures, antioxidant diets, have a significant probability of preventing these disorders. And what's nice about the dietary countermeasures, it doesn't need FDA approval. I'd like to note that the work here has really been, Jim and Barbara have been co-PIs on NASA grants since, I believe, 1992. We've had collaborators, Walter Hunt, who was with us at AFRI for a while, then Natalie here, Gemma Casadesus has been a significant contributor. Currently it's Shabu at HNRCA. One of my colleagues, Stephen Pitts at UMBC. Laboratory support has involved a significant number of people from HNRCA, some of my students at UMBC, and I apologize that I haven't, there just wasn't room on the slide to indicate everybody from HNRCA who has, in fact, been a significant contributor. So I thank you all very much for coming and listening to me.
If there is any morphological changes in the brain in which segment of the brain, and what kind of changes?
We have, or at least one of our collaborators, if we could ever get him to publish the data, has seen changes in dendritic morphology in the hippocampus and I believe in striatum and probably frontal cortex. The extent to which these are in fact related to the behavioral changes, I think, remains to be established. So yes, there are morphological changes in that regard.
Loss of cells? Or?
There has been, we cannot confirm the loss of cells. We did try some silver staining. I have to beat the person who's got this preliminary data on that. There does not appear to be a loss of cells, but we can't confirm that at the present time.
So it's the function of the cell?
It seems to be primarily functional, not in terms of the loss of cells, which is why I think microlesions are probably not the answer, direct hits on the cells are probably not the answer, but oxidative stress and how it affects neuronal function is probably what's going on.
So in one of the slides, you showed testing 11 months after exposure, and the slide said 8 weeks of diet intervention. So is that 8 weeks of diet intervention done immediately preceding the testing?
Yes. After, it's done just preceding the irradiation. Rats are then, that I showed here, were shipped back to UMBC and then were put on a normal diet. So it's prevention. Would be interesting if we can ever do it again to see whether it would be a post-irradiation type of thing. But we don't have that data yet.
So the other question has to do with, I guess it's your group work with that mTOR and sequestration 1. So those are all, both are involved in longevity, survivorship, various transgenic models. You can make mice longer live, you do one thing or another with it. But in the particular model that you're using, you've had such a very dramatic change, particularly in the sequestration 1. And those are, both are also involved in the general antioxidant pathway, the NRF1 and KEEP. And it would suggest that if you did other antioxidant interventions, that you would get a similar result. So the question is, has that been done? In other words, if it's not the food, is it just a general antioxidant effect?
There has been no attempt to look at mTOR or phosphorylation of tau using other antioxidant diets. There have been several studies that have used other antioxidant diets looking at other endpoints. So vitamin E, for example, has been reported to be effective for some of these endpoints, carcinogenesis or what have you. I have data I have shown here that it's also effective against carcinogenesis, adds to longevity, as well as blueberry and strawberry diets. So the suggestion is that it would probably be the antioxidant diet per se. Unfortunately, most of the data, unfortunately, is ours. And so we're limited by the behaviors we do and the physiological neuronal measures we do. But I would guess that the one thing our data has shown is that strawberry and blueberry are not always identical, at least in terms of their behavioral effects, whether it's striatal or hippocampal. So whether you would see a similar thing with other antioxidant diets is anybody's guess. Nobody has ever tried.
Are there any other questions? we should go to lunch now?